Today we have Marc Fuchs, a professor from Cornell University, and he will be presenting on updates on red blotch disease ecology. Welcome, Marc.
Thank you very much, Cindy. Thank you, the North Coast IPM program, for this wonderful and kind invite. I'm happy to be with you here and share the knowledge, and I have to admit the limited knowledge that I have on red blotch spread and disease ecology, but I'm happy to share with you everything that I know. And I thank each and every one of you for participating today. And I promise I will leave ample time at the end of my presentation for questions and answers, because I would love this presentation to be as much interactive as possible. So today we'll be talking about red blotch disease ecology, focusing on spread in the vineyard and how transmission actually occurs. And this type of talk, I think, is very timely in 2021, because believe it or not, I don't know how many of you realize that 2021 is the 10th anniversary of the discovery of grapevine red blotch virus from infected vines. So the virus has been identified 10 years ago, and there's still so many unknowns and gaps in knowledge that require more research activities. But anyway, today I'd like to offer to answer 4 questions. How is grapevine red blotch virus spreading in the vineyard? And up front, I have to say that this virus is the causal agent of the disease. The second question I'd like to address today is, how does the 3-corner alfalfa hopper transmit the virus, the causal agent of the disease? And up front, I have to make sure you understand that I refer to the 3-corner alfalfa hopper as a vector of the virus, not the vector of the virus. Why this distinction? Because I'm not claiming that the 3-corner alfalfa hopper is the only vector, insect vector, capable of spreading the virus in vineyard settings. The third question I'm going to address today and discuss with you are free-living vines serving as red blotch reservoir. You may know that outside vineyard settings, the only other host of the virus that has been identified so far are what I refer to as free-living vines, of vines that are capable of growing without any human intervention, whether being close to vineyard settings or far away from vineyard settings, in riparian area, in ditches, whatever, you name it. So wild grapes, or as I like to refer to them, free-living vines. And finally, what have we learned in terms of spread in the vineyard, transmission mode of the virus by the 3-cornered alfalfa hopper? How do these free-living vines contribute, if at all, in the epidemiology of the disease? And how does this information, the lessons we learn from this work, help us better inform disease management strategies? Okay, before I delve into the details of my presentation, I'd like to thank the people in my program. They are really amazing, and I'd like to recognize particularly 3 graduate students: Tori Hoyle, who is a 3rd-year grad student in my program; Maddie Flasco, a 4th-year grad student in my program here; and Libby Sienowitz, who used to be a grad student and graduated 3 years ago and moved on to a faculty position at Clemson University. So the first question is, how is red blotch spreading in the vineyard? We know that red blotch can spread in certain vineyards. It does not spread in other vineyards. But what do we know in terms of the spread in space and in time, and what have we learned? So up front, I have to tell you that on the East Coast of the US, although red blotch is widespread, it does not move from a diseased vine to a healthy vine, and it does not move from vineyard to vineyard. suggesting eventually that the vector does not exist in vineyards, or another alternative is maybe the vineyard managers might be doing a heck of a job at controlling these insect vectors. Anyway, no spread on the East Coast, but spread is documented on the North Coast and Northern California and in Oregon so far. So what I'd like to do is spend the next few minutes on 3 adjacent vineyard parcels in the Rutherford area in Napa, across the hill there from where you are. So I'm going to focus on a Cab Franc vineyard, a Cab Sauv, and a Merlot. And we're going to start with a Cab Franc, established in 2008, 5-acre block. And the vineyard manager only noticed disease symptoms in the 4th leaf in 2012. And I was fortunate enough to visit this Cab Franc block, and it called my attention because at the edge of the vineyard, close to the Napa River here, to the riparian area, there was a clear aggregation of diseased vines. So samples were collected from this area, and the presence of the virus was confirmed in 2013. So in 2014, we started to monitor the distribution vine by vine of diseased vines in that block from 2014 to 2016. And just to tell you, to show you how this aggregation of vines looked like, and there were about 20 to 25 vines about across 10 rows at the edge of the block. I will position myself here where you see the laser pointer with the buck facing the riparian area and the camera facing along the row. And this is what I had observed. Basically, almost every vine from the edge of the vineyard within about 20-25 vines was symptomatic, and suddenly all the other vines were asymptomatic. And I was sure that this would suggest that something was emanating from the riparian area and transmitting the virus throughout the block, particularly here. And I always, when you listen to me, I'm very proud of the grad students in my program, particularly when they prove me wrong. And obviously their work on virus spread in this block proved me wrong. And I will explain you shortly why. So we started to map the distribution of disease vines in 2014. And this is every cell on the top graph is represented the Cab Franc vineyard, the 5-acre one. Every cell is a vine. A red cell is a disease vine. A blank cell is a vine that does not show disease symptoms. And around the aggregated area here, I have arbitrarily delineated that square or rectangle to see how disease is evolving over time. The bottom graph is the same 5-acre Cab Franc vineyard for which the disease incidence is represented across the vineyard like this in panels of 5 vines. So in 2014, the incidence of red blotch was 4% throughout the whole block. But in the sub-portion that is arbitrarily delineated here, it is 26%. So in 2014, any diseased vine was highlighted in red. 2015 in green. 2016 in blue. And you can start seeing how this area is going to be filled up over time. Purple, 2017. Now we are in 2018. Almost every vine in that area is symptomatic. And more so, the disease is starting to go away, spreading within the vineyard away from the edge. 2019, almost no significant substantial increase. However, 2020, huge increase again, followed by 2021. So if I summarize what I have learned by monitoring that Cab Franc parcel over 8 years, and there is distinctly two different dynamics of spread. One that is predominant consists of random locations of newly diseased vines. So it seems here that if I'm a healthy vine, the likelihood of me becoming infected the next year is not influenced by whether I'm sitting close to healthy vines or diseased vines. It's a random distribution over time. This is in contrast to what is happening on the right-hand side, so the east side of the vineyard, where spread was definitely influenced by this initial aggregation of diseased vines. And when I look at the rate of transmission on the annual basis, there is a 1%, sometimes 2%, but roughly an average of an annual 1% increase in the number of infected vines on the west side of the vineyard. But on the east side, it varies from 10 to close to 20% in terms of annual increase. This is substantial. And believe it or not, despite my repeated attempts to convince the vineyard manager to rip out this area in terms of management, I was not very successful at delivering a resonating message. But lucky me, I'm able to continue monitoring the spread of this virus of concern in this particular vineyard. So now we talked about Cabernet Franc. Let me take you to the adjacent Cabernet Sauvignon, which is just across the dirt road of this Cab Franc. Again, 3-acre block, planted the same year as Cab Franc, 2008. But there, and like in the Cab Franc, where it took 4 years for the vineyard manager to notice disease symptoms, it just took 1 year. In the first leaf, he noticed red blotch disease symptoms. Now, when you look at this map, I'm pretty sure that some of you, unfortunately, experienced a similar situation that is illustrating the fact that the Cab Sauv material was sourced from 2 distinct nurseries. The one here, where the material was clean, and the one here, where the material was infected. So, and we confirmed, like for the Cab Franc, the presence of red blotch in 2013. So what is quite interesting here is, again, if I project myself in time and go back to the time these vineyards were planted. And obviously I'm speculating here because I was not present when these vineyards were established. I estimate that at the time of establishment in 2008, about 40% on the whole here of the vines in the Cab Sauv were infected with red blotch, versus here the aggregated area, which represents less than 1% of the Cab Franc parcel. Now, what is odd is the rate of virus transmission. It is barely occurring here, but we have a huge source of virus inoculum. So there is very little transmission from the segment of the Cab Sauv parcel that is infected to the healthy portion of the vineyard. Versus here, there is a little aggregation, but spread is rapidly occurring. How can we explain that? So what we did is we put sticky cards in that section of the vineyard and in that section with the idea that maybe the population of 3-cornered alfalfa hoppers are distinct and might explain the difference in dynamics of virus spread. And here, our hypothesis was actually validated because in the Cab Franc, we found 10-fold more 3-cornered alfalfa hopper insects than here. So what we learn here is spread dynamics is directly associated with the density of the insect vector population. Now, the last block where I study disease epidemiology is an adjacent Merlot. And this Merlot, let me go back to the previous slide, sorry, is hatched, showing full red. This is because almost every vine in that old Merlot was infected with Pierce's disease, fan leaf, red blotch, and leaf fall. So this long-awaited replant finally occurred in 2015 after the vineyard manager decided to rip out this filthy, for lack of a better term, Merlot. And we monitored since the time of establishment the incidence of red blotch. And not surprisingly, in the long-awaited arrival of red blotch was apparent in the third leaf. And we confirmed the presence of red blotch. And this is the distribution of the infected vines that we notice in the third leaf. Again, an aggregation on this side of the block and then randomly distributed individually diseased vines, which obviously this aggregation had reminded me of what we had observed years prior in the Cap Franc. Now, you might know or recall that genetically speaking, when you look at red blotch virus, there are two distinct strains of the virus that can be identified. They're just simply called, they're not called Delta and Omicron, they're called simply 1 and 2. And the most prevalent strain in red blotch infected vineyards is strain 2. So what we found is strain 2 is predominant in the Cap Franc. In the Cap Sauv, we have a mixture of strains 1 and 2. And as I mentioned, a number of free-living vines in riparian areas, as indicated here, can also be infected with red blotch. And these particular free-living vines are infected with strain 2 of red blotch. So obviously we wanted to know which strain were present in or was present in the Merlot vineyard. And we had hypothesized that because strain 2 is prevalent, particularly in close proximity in the Cap Franc and in the free-living vines, that strain 2 would be present in the Merlot. And yes, we were right, but sort of right and sort of wrong as well, because the only vine that is infected with strain 2 of red blotch is the lonely vine here that very likely got infected after a vector visited it. All the other ones, and particularly here, in that aggregated area, are all infected with grapevine, with strain 1 of red blotch virus. So what does this indicate? How can we explain the provenance of strain 1 in an area that is surrounded by strain 2? Again, the planting material. Although this material was certified Protocol 2010 derived, it was infected with red blotch 1. So what did we learn by doing some monitoring over time in these 3 blocks? And I think there are some lessons, and you might have experienced similar situations in your estates. Is, to me, the planting material is by far the major source of red blotch in vineyards. And it remains by far, as we speak today, the major source of the virus in vineyards. What we have learned by doing epidemiology work is that spread occurs primarily from within the vineyard. In other words, if you have established diseased vines, this is where the virus will be spread out by the insect vectors, rather than an influx of virus from the outside within the vineyard of interest. I'm not saying that there is no influx from outside sources. I'm just saying that the primary occurrence of spread is explained from within the vineyard. And spread is slow, overall speaking, and that's for the majority of the vineyards, not only the ones that I showcased to you today, that those that I visited, except when there is a high density of the vector population, and more so when at the beginning of an epidemic, there is an aggregation of diseased vines. What I've learned also is there is some significant latency for symptom development to occur if the rootstock is the source of the virus. And this is what we have seen actually in the Cab Franc in both the Merlot, because the vineyard manager in the Cab Franc, it took him 4 years to see disease symptoms. And that is a clear indication that it takes 3 to 4 years for the virus to translocate from a rootstock into the sign, build up into the sign, and then disease symptoms developing. And as I mentioned, aggregation may contribute to the fast spread. And this is obviously something to avoid in order to mitigate the impact of red blotch. And as illustrated, the abundance of the 3 Cornell alfalfa population correlates with spread dynamics. So I mentioned a lot about the 3 Cornell alfalfa hopper. How does it transmit the virus? And we had little information when we started this work, except we used similar viruses and made by analogy an assumption. And you know, I have to apologize for my poor drawing skills. The image in the middle is supposed to represent a 3 Cornell alfalfa hopper, and the real specimen is shown on the right inset there. But anyhow, I'm going to use this cartoon representation to explain how the virus is acquired by this insect vector, at least how we hypothesize that transmission is occurring. So when a 3 Cornell alfalfa hopper feeds on a vine, and I think we all agree that this insect species is not a pest of grapes, and we'll come back to that notion a bit later, it will feed on the phloem sap. And it does that by using a stylet that inserts into the layers of the tissue it feeds on, primarily petioles, shoots, rachises, and so forth, and inserts it where actually the virus is located, in the phloem cells. And by absorbing the sap through the food canal, it will acquire the virus. But we are all familiar, at least most of us, to how leaf roll is acquired by mealybugs. And mealybugs will feed in a similar manner than the 3 Cornell alfalfa hopper, except mealybugs, when they are acquired, they will stay within the stylet to that area. This is not the case for red blotch, because red blotch, after it is acquired through a feeding probe of the insect, will move up the canal, the food canal, and transit through the insect body. So we go through the foregut, the hindgut, the midgut, and then the hindgut. And once the virus has transited to these tissues, it will exit the gut epithelium and enter the hemocoel, or the hemolymph of the insect, which is the equivalent of the blood for human beings and animals. Once it is in the hemolymph, the virus can circulate within the whole body of the insect. And particularly important is the transition from the hemolymph into the salivary glands. And the salivary glands are located behind the eyeballs, if you want, roughly speaking, of this 3-cornered alfalfa hopper. And going into the salivary gland is hypothesized to be critical for the virus to then be ejected from the insect body through the salivary canal during another feeding probe on a healthy vine. And then the virus particles are released in the phloem sap and infection can start. So this is what we hypothesized. So the virus has to enter and circulate within the body of the insect vector. And therefore, we hypothesized what is referred to this circulative transmission mode. So what is critical, again, once you expose an insect vector to an infected vine, if you want to study the transmission, is to make sure that we work with virus present in the salivary gland, not in the gut. Because if the virus is in the gut, it cannot be transmitted and start a new infection on a healthy plant. The virus has to be in the gut. So how can we distinguish the virus in the salivary glands from the gut? And it's very important because once you expose an insect to an infected vine, you expect it to acquire the virus and transmit it. So what we did, we took advantage of different plant species in our transmission assays. We use grapes, obviously, and we know that grape is a host of red blotch virus. We know that grape is also a feeding host thanks to the work that Cindy and colleagues have done a while back, but it's not a good feeding host. Actually, the 3-cornered alfalfa hopper does not like grapes, put bluntly. Actually, it even hates grapes. It rather dies than to survive and reproduce on grapes. And I will come back to that later. Then we took advantage of alfalfa because you might know that leguminous species are preferred feeding hosts and reproductive hosts of the 3-cornered alfalfa hopper. What is interesting, again, alfalfa is a fantastic feeding host, but is not a host of the virus. And we come back to why alfalfa is important in our transmission assays. And then the last one that we used is snap bean in our transmission assay because it is a host of the virus and the insect vector. So just to illustrate how important it is to use different plant species if one is interested in doing meaningful transmission assays, I just show you results of several experiments where we exposed the 3-cornered alfalfa hopper to infected vines for several days and tested them over time. And roughly, we get about 80% when we test the entire insect, 80% of them that carry the virus. But then, after we expose, again, a population of insects for extended time on an infected vine, and move them to alfalfa for extended days again, to clear their guts. In other words, the insect that will acquire the virus will continue feeding from grapes when we put them on alfalfa, but all the virus load in the gut will get ejected, and only those that have the virus in the salivary glands will be detected. So, and this is what happens here, where you see that the proportion of insects that were exposed and tested positive for virus dropped basically from 80% to 40%. So that's also one lesson that we learn is it's not because an insect feeds on a vine that it is able to acquire the virus and for the virus to be transmissible. Because again, what is really critical is the virus needs to be in the salivary glands to be able to be transmitted. So then the next step is we wanted to detect the virus in the body of the insect. We dissected the different organs. We dissected salivary glands, hemophil, hemolymph, hemocele, and gut. And we detected, and we checked how long it would take for the virus to be detected in these different organs after the insects were exposed to infected vines, and they spent 2 days for gut clearing on alfalfa. And this table, I know it's very busy, but it's indicating that it takes 5 days of exposure of the insects to infected vines for the virus to be detected in the gut, 8 days for the virus to be detected in hemolymph, and 10 days for the virus to be detected in the salivary glands. And the virus can only be transmitted at 10 and more days of exposure to infected vine. The virus is not transmissible if it is not present in the head and in the salivary glands, as we have documented, but I will not show you the data at this point in time. The other question that we try to address to better understanding how this interaction of the virus and the 3-cornered alfalfa hopper is occurring, the question was, is the insect, we know that the grape, whether cultivated or free-living grapes, are hosts of the virus. Is the 3-cornered alfalfa hopper a host of the virus? In other words, can the virus multiply in the insect vector? And the answer is no, because when we quantify the amount of virus present in the 3-cornered alfalfa hopper specimens that I fed for 0, 3, 6, 9, and 12 days on grapes and spend so much time on alfalfa, you can see that the amount of virus is decreasing over time. So the virus is just hitchhiking within the 3-cornered alfalfa hopper in order to move from an infected vine to a healthy vine. It does not use the insect vector as a host. So let's talk more a little bit about transmission efficacy. So we established these transmission assays where we exposed this 3-cornered alfalfa insects for at least 2 weeks on infected vines, move them for at least 2 days on alfalfa, and then we use as recipients excised grape leaves. And why did we use excised grape leaves instead of entire vines as recipients here is because we wanted to restrict the feeding of the insects in a tissue that is of a size manageable to detect the virus. In other words, we wanted to increase our chances to detect the presence of the virus. That's why we use excised leaves instead of entire potted vines. And this is a photograph showing how this assay is developed. And you see after the transmission assay, the alfalfa hoppers on the excised grape leaf. So this is the rate of transmission that we obtained. It is pretty low. So in other words, our transmission assays conducted in a confined environment in the lab, in other words, is basically confirming that spread does not occur very efficiently in a vineyard, with the exception that I mentioned, particularly when there is an area of the vineyard where the vines are aggregated, the disease vines are aggregated. What is interesting in our assays, and I mentioned a few minutes ago that grape is a feeding host, but a very poor feeding host of the vector, but leguminous plants like snap bean are preferred hosts. So when I do the assay using infected grape or infected beans, you can see here the difference in the transmission rate. And it's not because there is more virus in grapes and in beans. It is just the difference between 10 and 100% is explained by the behavior of the 3-cornered alfalfa hopper on grape or beans. They just love beans. They cannot stop feeding on beans if I cage them. If I cage the hoppers on grapes, they spend more time on the walls of the grapes than on the vine itself. So again, you see the similar trend. We have higher transmission efficiency if instead of a detached grape leaf, I use a detached bean leaf. So what have we learned? That transmission is occurring only if the virus is transits through the insect body, but the virus uses the insect just as a hitchhiking vehicle to move from an infected vine to a healthy vine. It does not multiply within the insects. And at least 10 days of exposure to an infected vine are needed for the hopper to acquire the virus. Now, again, I have to put this in a broader context because 10 days of exposure is in a greenhouse setting where I cage the insects with an infected vine. Obviously, this has little bearing on a vineyard situation because the hoppers are not caged. They can move freely, and I'm pretty convinced that by hopping from infected vine to infected vine, more so if in an aggregated area you have many diseased vines, there is ample time for the virus to circulate much faster than 10 days as documented here, move faster within the body of the insect than the 10 days shown here. And transmission of the virus from wine grape to wine grape is not efficient whatsoever. So Cindy and colleagues also showed that, as I mentioned several times, they elegantly showed that grape is not a reproductive host of the 3-corner alfalfa hopper. In other words, the adults cannot they will reproduce, oviposition will occur, eggs will hatch, first, second, and third instars will develop on wine grapes, but then the cycle is interrupted. There is no fourth and fifth instars, and like the cycle that you see here from egg to adult that is occurring on alfalfa within 50 to 60 days, roughly. So knowing that and acknowledging that oviposition can occur on grapes, eggs can hatch, first, second, and third instars can develop. The question was, if they were to develop an infected grape, could the first, second, and third instars acquire the virus? And if yes, would the progeny carry the virus? So here you see how this reproduction cycle is occurring and how molting is occurring, because you see here one exoskeleton, and I would venture to say that this is a fourth or very likely a fifth instar that is just exiting the exoskeleton. So the idea was very simple. We let the alfalfa hoppers feed on an infected vine, and as soon as we notice the first instars develop, or the second, or the third, move them to beans, to healthy beans. And we continue monitoring them, the first to second to third to fourth to fifth, and to adults. And we did the same with the second instars that develop on grapes, third instars that develop on grapes. So keep in mind, the adults deriving from the first, second, and third instars, they developed on healthy beans. They have never been exposed to an infected grape during their livelihood, okay? So we tested these adults to see how many did carry the virus. And to our big amaze, all of them, whether derived from first, second, or third instars, all of them contain the virus. So indicating that transmission is occurring across the different development stages of the insect vector, and it's referred to as a transstadial transmission. Again, this has profound implication in terms of epidemiology, because these adults were never exposed to any virus-infected plant, okay? The instarts they derived from were exposed, but not the adults. So what have we learned? Transmission is transstadial. What we also documented is adults are more efficient vectors compared to nymphs. So this is contrary to what is happening with mealybugs and leaf roll. And what we also learned is once an adult has acquired the virus, he can retain it for more than 30 days. And we are still fine-tuning these 30 days because our latest experiment showed that they carry it beyond 40 days. And if they carry it for more than 30 or 40 days, it means that they can inoculate and infect plants during that very, very long time period. So what's happening with the transmission in the vineyard? What do we know? Is the 3-corner alfalfa hopper a vector of red blotch in a vineyard? So we started an experiment 2 years ago during the pandemic in New York, and we selected an isolated Cap Franc vineyard planted in 2018. And just as a reminder, there is no 3-corner alfalfa hopper that is known to occur in New York vineyards. So that's why we wanted to do these experiments under controlled conditions, semi-controlled conditions, but in an isolated field with no alfalfa field close by. So what we did was very simple. We exposed 3-corner alfalfa hoppers either to healthy or infected vines. We carried them, let them starve for a couple of hours prior to their release on shoots of this healthy Cap Franc vineyard. And after 4 to 5 weeks, we applied a contact insecticide to make sure that any survival insects would be killed before opening these sleeves in which they were contained for 4 to 5 weeks, so we could monitor feeding damage and the presence of the virus over time. And we also obviously monitored the insect behavior in these sleeves over time. So the first thing we measured was feeding. And as you can see here on the left, by these white arrows, you recognize the feeding damage and the girdling of the petioles that has extensively occurred. And you can distinguish also some reddening, interveinal reddening, veinal reddening that was observed in some of these sleeves. So again, keep in mind, we had insects that did carry the virus. We referred them as to viriliferous, or insects that did not carry the virus, or aviriliferous. And here you see just a quick graph representation on the feeding damage. There was no difference in what we observed if the shoots were exposed to insects carrying or not the virus. In terms of leaf reddening, again, there was no statistically significant difference in terms of the number of red leaves, whether the shoots were exposed to insects carrying the virus or not carrying the virus. So again, just to illustrate how poor of a host grape is to the 3-corner alfalfa hopper, you can see here the survival, or actually the rate of mortality of these insects in the sleeves over time, over the 4 to 5 week period of the experiment. And you can see that at 4 weeks, you have almost every specimen that was caged, if you want, in these plastic sleeves that were dead. And even 1 week after the release, about 30% were already dead. Again, illustrating the fact that the 3-corner alfalfa hopper will feed on grapes, but really does not like grapes. It rather starves when exposed to grapes and die than to feed on grapes. But more importantly, 3 months after the exposure of the vines to the insects, we had some vines, or some shoots, I should say, that tested positive. And none of the controls tested positive as expected. So we continue monitoring these vines to see how the virus from the shoots that was caged is moving throughout the vine, and how disease symptoms, red blotch disease symptoms, evolve over time. And I can tell you, I'm still waiting, as we speak today, for these infected vines to show disease symptoms. That has been more than a year after we detected the presence of the virus in these shoots. So to wrap up very quickly, the last question I wanted to address, we know that free-living vines carry the virus. And some of these vines grow in riparian areas here. So the question was, if the virus is present in free-living vines, can the 3-cornered alpha hopper transmit it from an infected free-living vine, again with the alfalfa to clear the gut, into the touch leaves of wild vines? And when I talk about free-living wild vines, I'm referring to Vitis californica or hybrids of Vitis californica and wine grapes, or hybrids of Vitis californica and rootstocks that are widely present in Northern California. And what we have seen here is a very high efficiency of transmission from wild grape to wild grape. And you remember, we had only 10% from cultivated or wine grapes to wine grapes, but from free-living vine to free-living vine, the transmission is very efficient. The transmission from free-living vine to a wine grape is occurring, but again, at a much lower rate. And the transmission is similarly occurring from a wine grape to a free-living grape with, again, a higher efficiency. So what we have seen is transmission occurs to and from free-living vines. Transmission is very high from free-living to free-living vines. And we try to understand why there would be such a differential transmission rate, again, in light of the behavior of the 3-cornered alfalfa hopper and this distinct behavior on wild or free-living vines versus wine grapes. And what we found is free-living vines are actually not only excellent feeding hosts of the 3-cornered alfalfa hopper, but also reproductive hosts. In other words, the reproductive cycle that I had illustrated on alfalfa is a reality on free-living vines. So what did we learn in terms of disease ecology? We learned that the 3-cornered alfalfa hopper is indeed a vector of red blotch and the greenhouse conditions and in the vineyard. Both adults and nymphs are vectors capable of acquiring and transmitting the virus, unfortunately, but adults are much more efficient vectors than nymphs. The virus has to transit within the body of the insect, but does not propagate within the insect. Importantly, the virus persists in the 3-cornered alfalfa hopper through molting. So it means that it is conceivable that the population of insects naturally occurs in different landscapes, with these insects never being exposed to a single virus-infected vine. And again, all the work is in progress. So far, we could convincingly conclude that the insect vector can retain the virus for at least 30 days, if not more. Transmission from vine grape to vine grape is very low. In contrast, transmission is very high from free-living to free-living vines. And as I just mentioned, free-living vines are feeding and reproductive hosts of the insect vector. And I will not expand, but there is no seed transmission of the virus whatsoever. So in wrapping up, how do these findings inform management decisions? I will not go through this slide in details. I will give you the freedom to read yourself. Basically, my point is I'm going to provide a message that is in line with the previous talks this morning. Do not worry about the 3 Cornell alfalfa hopper. In other words, do not apply any insecticides to kill that virus population. This insect vector is not a pest, is a poor transmitter of the virus. Don't worry about the insect vector. Put your emphasis on the virus inoculum, on the disease vines. Remove these disease vines if you document spread. If unfortunately you deal with an aggregated area that will speed up the rate of spread to a 20% annual increase, and if you're inclined to do so, contact your California Department of Fish and Wildlife, get a permit, and if you worry about free-living vines nearby because they serve as a source of inoculum that will come to your vineyards, try to remove these free-living vines. And more importantly, the lesson that I learned being involved for Red Blotch in 10 years, still, as we speak today, the planting material is the major concern and the major source of the virus. With no further ado, I thank you and each and every one of you for your attention. I thank the funding agency that support my program and each and every one of you again, because you might not realize how impactful you are on my research program. And with no further ado, I know that Cindy is eager to cut me off and for me to entertain questions. Thank you.
Yes, thank you, Marc, for your presentation today. Michelle is going to read you. You have quite a few questions in the Q&A, so let's get started. Michelle's going to read you your questions.
Yeah, so you actually have 20 questions. The first one was, could we see pictures of the leaves that were deemed symptomatic? I think you showed some of those pictures in your presentation, but if you could go back and show them again.
The disease pictures, you mean?
Yes, of diseased leaves.
Let's see. Was it this disease leaves or the insect release experiment?
I think they were interested in what you considered symptomatic leaf symptoms. Which
Actually, yeah, okay, I'm sorry, now I'm with you. I did not consider symptomatic in terms of disease any of let me see. I think we are referring to this slide. Just bear with me. Oh, why doesn't he show? Sorry, going through. If we are referring to, oops, this one, right? Yes.
I would think so. There's more specification in the question.
Yeah, I'm sorry, I went through very quickly. None of those are diseased.
It's a response from girdling.
That is absolutely correct. It's a result of the insect feeding. It's not because the 3 pictures here were taken on shoots exposed to insect not carrying the virus.
Okay.
Got it. The next question is, was there any effort to reduce the spread of Cab Franc by way of prophylactic insecticide applications?
No, there was no attempt whatsoever. And again, my conclusion is I do not recommend any insecticide application to reduce the presence of the insects. because I want you guys to remain the best stewards of the land, and I would hate for you to waste your money.
Are you aware of any local labs that can provide us with Grbv strain?
Red
Blotch associated virus strain.
That's
I'm not
an unexpected question, actually, because you would not want to deal with a virus, right? And no, there is no one that I'm aware of that is even capable of producing the virus, because we have tools to detect it. We have tools to infect a healthy vine, but we are still struggling on developing reliable procedures to extract the virus from an infected vine and purify it. So I wouldn't be able to tell you a single lab capable of providing you with red blotch.
Does the virus express differently, latency, severity, etc., on different rootstocks?
The short answer is no, because rootstocks can readily be infected, but they are all latent, meaning they do not express any disease symptoms. But they can all be infected.
Got it. Can budding over substantially reduce virus?
You mean top grafting?
The question is, can budding over substantially reduce the virus?
So that'd be like chip budding, like field grafting.
No, the short answer is no. Once you have a vine that is infected with red blotch, it will remain infected as long as it is in the ground. The only way, and whether you top graft, you chip bud or whatever, the virus titer will be there. You might just delay the translocation of the virus from the piece that remains in the ground to the chip budded tissue. But the virus will move. It's just a matter of time.
Are locally systematic insecticides applied in correlation with trap counts an effective control mechanism?
Again, it's an excellent question, but I do not recommend the use of any insecticide, whether contact or systemic, to control this 3-corner alfalfa hopper. Just ignore them. Just pretend they are not there. Focus your efforts on the virus. Get rid of the virus. Don't worry about the insect.
Do you think Bavaria
species
fungi might infect attack TCAH?
The likelihood is pretty high, yes, if you find the right strain of Bavaria. Again, these are good thoughts, and I'm grateful to everyone to share them, but I'm going to repeat myself. Do not worry about these insects. They are cute. Yes, they are transmitting the virus. They're cute. They're lovely. Let them be. They hate grapes. They hate vineyards. Do not worry about them. Get rid of the virus. Make sure that when you establish a new vineyard, you spend tremendous time with your favorite nursery people and make sure that you establish clean vines.
The next question, would bell beans in vineyards increase transmission?
Theoretically, I think it is possible, right? Because a number of these leguminous species, particularly bell beans, that's the work, I'm sorry, Cindy, that again, I'm going to refer to your fantastic work, have been shown to be feeding a reproductive host, some preferential feeding a reproductive host, but some that are used in cover crop mixes definitely could potentially have a role. My philosophy is always, I'm hesitant to embrace blindly the fact that cover crop mixes have a role in the epidemiology of the disease, because in Northern California, there are the diversity of legume species in the landscapes is such that I'm questioning whether the three-cornered alfalfa hopper really needs to reproduce on cover crop mixes versus, oh, I'm happy enough to find everything I need in riparian areas.
Great. The next question is, are all legumes a host for Grbv, or are there any other plants?
So the short answer is no. I'm using snap beans just as a model host in the lab. What I have to mention, which I failed, and I have to apologize, we tested years ago over 3 consecutive growing seasons, a lot of cover crop species that are commonly used in Northern California to see whether any that are grown in diseased vineyards did test positive for the virus, including leguminous species, veg, trefoil, blank out on, there are many others. And none of them tested positive for the virus. None, absolutely none.
I received a clarification on that question earlier about the virus in the labs. The question was for a lab which could report on the clade of grapevine red blotch virus.
Oh, I'm sorry. Yeah, I misunderstood the question. I'm not aware of a lab offering this type of service. The question is, I think it's a good question because it has value in terms of helping conversations with neighbors, with nurseries, right? But in terms of the biology of the disease, we do not know any biological significance as to why there would be a strain 1 or a strain 2. But definitely as a marker to understand where an epidemic is starting and where it goes, yes, that would be useful. Absolutely correct.
So the next question, can you please provide the scientific name of snap pea? Is it, I'm not sure how to say that, I'm not gonna.
Phaseolus vulgaris. Phaseolus vulgaris, yes.
Okay.
It's the common bean.
Perfect. And then can some bean cover crops be a source of Grbv?
No. Again, we have done extensive tests. No, sorry, let me rephrase my reply here. Based on our experience, tested hundreds of cover crop species and samples, particularly leguminous species, we have not found a single one infected with the virus. Again, that doesn't mean that there are none out there, right? But I highly doubt that they are having any major role in the epidemiology of red blotch disease.
Great. Are residual roots from previous vineyards a source of Grbv for nematode spread to a new planting?
So there are 2 questions. Fruits, by definition, regardless of what you might consider as an insect vector, they will not be involved in any transmission whatsoever of red blotch. So you can exclude any role. Second, there is mentioning of a nematode spread. Red blotch is not transmitted by nematodes whatsoever. There is no indication that there is a soil-borne step in the cycle of the disease. The transmission is only aerial. And no, sorry, I should mention again, to reemphasize, human beings are the major spreader of the disease. Okay, keep this in mind. We are the best, we are much more efficient than the three-cornered alfalfa hopper at spreading red blotch. And as long as we don't put our acts together, we're gonna to continue suffering with this disease of concern.
Got it. This question is from someone who's asked a question before. They said, per my previous question, in vineyards, are legumes, where legumes have been used historically, and propagate spontaneously, would applying systematic insecticides across the understory at the right time be an effective strategy for control?
I think some of the follow-up speakers might address this. My philosophy, my approach, based on my experience, no hardcore research evidence would indicate that that might be a waste of time.
Thank you. Regarding transstadial transmission, could the first instar infect the clean snap bee virus replicate increase in bean, and third and fourth instar adult became infected? I'm sorry if that question, I'm not sure if the...
No, I think I follow up. The short answer is no. And we have evidence that it's no.
Great, thank you. We are, I think, in the break portion. Is that correct, Cindy?
Yes, so we are in the break portion. If you don't mind, Marc, we can continue reading questions during the break time. For those who would like to hear the answers live, for other people that need to take your break, now's the time to take a break, and we would start the program back at 10:15 in 9 minutes. So if that's good with you, Marc. I'm happy
to stay if you if
you have a lot of questions.
Yeah, a lot of questions.
So the next question sounds like the major problem is in the nursery. How can nurseries eliminate their spread?
Well, the first thing that I have to admit, and I think we all agree, is nurseries have gotten a wake-up call when we realized that red blotch was first of concern. When tools were available and they were extensively used, and realized that red blotch was everywhere in their increase blocks. So every nursery I'm aware of have done their due diligence in terms of really, destroying their previous increase blocks, reestablishing new ones in more or less isolated sites, and some have done a better job than others. But that's not enough. Testing has to be done, particularly on rootstocks, where, as said again, infection is latent. In other words, the virus can infect the stalks, but they will never show any symptoms. Similar to leaf roll. Rootstocks can be infected with leaf roll. They will never express any disease symptoms. And some have been doing their due diligence in terms of being much more aggressive in terms of testing. Some offer testing on the material that the growers are purchasing. What is really missing in the picture, and I'll be very blunt with everyone, is as long as every vine in an increased block is not tested and emphasis is put on the material sold, we are not solving any issue. So there is plenty of simple steps to be taken, and if the grower community doesn't ask for clean material, and when I mean clean, seriously clean, it will not happen. There needs to be a demand. If there is no demand, there is no action taken, and you're gonna to suffer, continue suffering.
Thank you. The next question, in the Cabernet Franc vineyard you studied, why would you guess that there was little spread of the virus in 2019?
That's a wonderful question. And if I had the answer, I'd be happy to share it. But so it's going to be a gut feeling type of reply. What we have to keep in mind that what we measure at a given time and what we observe is a result of a sequence of events that happened in the past, right? So what I have observed in 19 is a result of what happened prior to 19. What we don't know at this point in time, because we don't know what the incubation time is of the virus, once a 3-corneal alfalfa hopper carrying the virus has visited that vine, how long does it take for the virus to multiply that vine, build up to a certain level so that disease symptom expressing? So what I have observed is very frustrating to me because I don't know if it's the result of something that happened 18, 17, 16. I just don't know. If I had to guess, I would say it's what I observed in 19 resulted for something happening 2 or 3, if not 4 years prior. And there I don't have a good record, and I failed to document the insect population. I failed to document, weather conditions and so forth. So it's very difficult for me to guess what might have happened.
Thank you. The next question is, can you elaborate on free living vines? Do you know what species or mix of species they are?
Yes, I do. The ones that I used in the work that I presented during my presentation this morning were hybrids between Vitis californica and Sauvignon Blanc. And I know that because at Foundation Plant Services, there is a unit led by Bud Dangle that is capable of genotyping the free-wheeling vines or any cultivated cultivar, clone, or rootstock. So in working with them, they genotype the material that I use for the transmission assays as a hybrid between Californica and Sauvignon Blanc.
Great. The next question, can you explain again the 5-5 transmission trial from bean to bean? Does that mean beans carry the virus, or just that beans do not help clear the gut?
So the bean was used, let me backtrack a little bit. We have done, like many colleagues, grape-to-grape transmissions, right? And most of us have failed to successfully transmit red blotch, or the transmission rates were so low that it was very frustrating. Although I had this real strong conviction that the 3 Cornell alfalfa hopper was actually one of the vectors, of the vector. So I wanted to explore with the grad students other hosts that could be used as a landmark to have high transmission. So with this idea in mind, and to put the low transmission from grape to grape in a bigger perspective, with this in mind, we explored leguminous species because we know they're the preferred hosts and reproductive hosts of the 3 Cornell alfalfa hopper. And we tried artificially in the lab to inoculate those. And very early on, we quickly realized among all the species that we tested, the common bean was the one that could be artificially inoculated with the virus, could sustain the virus infection, and knowing that the insects are very happy on beans, they acquired the virus very readily, very quickly, and transmitted it very happily on snap beans. The gut clearing, for the gut clearing and making sure that the virus is in the salivary glands and head, we used alfalfa. Why did we use alfalfa? Alfalfa is, again, a leguminous species, preferred host and reproductive host of the insect, but it cannot be infected with the virus, okay? So by exposing the insects, they will feed, they will clear their gut, but the virus will not be transmitted.
Great, thank you. We have about 1 minute left. So I think you still have 21 open questions, but I'll just ask this question. Can the virus exist at undetectable titer levels in nursery materials?
Yes.
And then,
Especially,
how-
I'm gonna to, yes, especially if the wrong tissue in the wrong time of the year, the test is run at the wrong time of the year. Yes, absolutely.
great.
Okay, so I'm sorry, we're at 10:15. We have to start on for the next presentation.